2008, it is estimated that 1 in 8 women will develop breast cancer over the course of her lifetime. Breast cancer is the second most common cancer diagnosed in women in the United States, and the occurrence of the disease has once again begun increasing. The most severe form of breast cancer is metastatic cancer, and this type of cancer spreads from the breast tissue to a secondary location. According to Mundy and Rubens in their book, Cancer and the Skeleton, the most common secondary location is bone, and once these cancer cells colonize bone, the cure rate is almost zero. My interest in breast cancer led me to join a lab group last spring whose research studies cancer that spreads to bone. And so for the past 6 months, I have been researching treatment options on breast cancer cells and bone cells in vitro, or outside of the body in tissue culture. My experiments, however, have been extremely frustrating because of the limitations of cell culture methods. Conventional cell culture systems are simply not complex enough to model this involved process. So throughout this presentation, I'd like to explain to you some of the major flaws of conventional methods, but first I'd like to talk about what actually happens when cancer spreads to bone. So the process by which cancer metastasizes is very complex. First, cancer cells need to leave the breast tissue and travel to the bone, where they then interact with the different cell types there. Your bone is a very dynamic tissue. It is constantly being remodeled. And there are 2 cell types responsible for this remodeling process. The first of which is the osteoblasts. Osteoblasts are bone-forming cells. They deposit the matrix that actually hardens bone. The other prevalent cell is the osteoclast, which has the reverse action. It degrades bone. So in a healthy individual, the activity of the osteoblasts and osteoclasts are at an equilibrium. However, when cancer cells challenge the bone, they disrupt this balance. Cancer cells will interact with the osteoblasts to limit their function. But on the other hand, they actually increase osteoclast activity. So the result is a lot of bone degradation, and this causes the weak and brittle bones found in many cancer patients. So to study this disease, it's necessary to take these cells, bone cells, and cancer cells, and place them in a tissue culture system. However, when you do that, when you take a cell out of its home in the body, you subject it to sudden and drastic changes in environment. In a review in Cell by Scher and DePino, they refer to this adaptive process as culture shock. And there are a lot of factors that contribute to culture shock. Too many for me to go into in this presentation, but the main point is that a culture system should try to mediate those effects. However, conventional cell culture systems seem to amplify culture shock, and they do that by creating an extremely unstable cellular environment. So I've created this image to sort of show you the basics of cell culture methods. So first, you would take the cell out of the body and deposit it on a layer of a certain type of plastic in a culture dish, and you allow the cells to attach to that plastic. Then you supply the cell with a fluid that contains nutrients. We refer to that as medium, and that's shown in pink here. So as cells grow, they take in nutrients from the medium, and then they produce waste products. In addition to these waste molecules, they produce large proteins that we refer to as macromolecules. So to allow these cells to grow long term, you need to provide them with more nutrients and get rid of those waste products. And when you do that, you change the media. However, when you change the media, you remove everything, including those macromolecules that the cells have produced. Unfortunately, for bone cells, these macromolecules are necessary for bone mineralization or hardening. So to date, no one has been able to produce a hard bone sample in these methods. So as you've seen, there are many instabilities associated with standard tissue cultures, one being this concept of removing the macromolecules, and the other being the immense amount of culture shock a cell can go through. So this suggests that we need a better in vitro model to study cancer metastasis to bone. This year, approximately 40,480 women are expected to die from breast cancer. And I believe if we put our time and resources into studying this disease and developing a method, we could find a treatment. Thank you.